KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.com.
Students, now in this video we are discussing primary and secondary metabolites. Okay, so in this video we are discussing regarding primary and secondary metabolites. So what is the primary metabolites and what are the uses of primary metabolites? Later, what is the secondary metabolites and what are the uses of secondary metabolites? After completion of that particular small concept and introduction information of biomacromolecules. So somewhat information also we will concentrate. Let us start to move forward to discuss regarding this one. So in the chapter of biomolecules now, so initially we have discussed the introduction, later we have discussed regarding amino acids, and later we have discussed on regarding which one, like lipids. Later we have discussed regarding DNA and RNA, what are the macromolecules or what are the components or present in the nucleic acids we have been discussed. Now in this video, purely and perfectly we will discuss regarding primary and secondary metabolites. Okay, now let us see the most exciting aspect of chemistry. So chemistry point of view or a chemistry subject point of view, most exciting aspect is that. So what is the main or most exciting topic now, which deals with the isolating thousands of compounds, small and big, from living organisms. So means here by using of chemistry or the chemistry technological information, so here definitely we can isolate the thousands of compounds. So what are the components which are present in the living organisms? So definitely we can isolate. So that are maybe, so these compounds may be small and big from the living organisms. Means from the living organisms we can isolate small and big kind of compounds. So or we can isolate thousands of compounds that are maybe small or that are maybe big one. From where we can isolate? From the living organisms, isn't it? So simple, so by using of chemistry, just we are isolating the thousands of molecules that are maybe small or that are maybe big, whatever it is, but definitely we are isolating. Suppose if you see there, suppose if you have leaf in your hand, no, so what are the components or what are the pigments or what are the chemistry or what are the compounds are present in the leaves? Very easily we can find out. Okay, so now here determining their structure and if possibility synthesizing them. So here when we are isolating the thousands of compounds from the living organisms, no, so after that one we are determining their structure. So definitely we are studying that particular structure and we are learning that particular structure. Not only that one, if a possibility is there, we are synthesizing also them. So we are ready to synthesize them in our laboratory conditions. Point purida, so simple. Initially these 4 lines, what is the point we learn? No, by using of chemistry subject we can isolate the what are the chemicals present in the both plants and living organisms, we can isolate. So if you isolate means thousands of compounds we will obtain that are maybe small or that are maybe big. Okay, so and once that particular isolation is completed, so we can determine their structure. we can learn the particular structure, and if a possibility is there, we will synthesize them in our laboratory conditions. So means by providing of suitable conditions, so definitely we can synthesize them in laboratory conditions. So that is the point we are discussing. Okay, now see there, if one were to make a list of biomolecules, such a list would have thousands of organic compounds including amino acids, sugars. You see there, likewise, suppose if you prepare one list, if you make one list of biomolecules, so that particular list would have thousands of organic compounds. Already we have discussed this one. Suppose whatever may be that living organisms or tissue, if you take it out, and if one movie, suppose English movie nalli, so they are taking one what is a cake. So for example, but there while checking and all, policemen, they will keep some kind of mission into the cake, so there they will give the information, okay, carbohydrates this much, proteins content this much, or this one, so on. So likewise, one organic compounds list, or what are the chemicals, or what are the proteins, carbohydrates, vitamins they have used for the preparation of cake, each and every point, minute detail also it will come out. So likewise, likewise, suppose here make a list of biomolecules. Suppose if you are taking from the, or a list if you would like to obtain from the any kind of living organism, so at the time thousands of organic compounds we have to list it out, including amino acids, sugars, etc. So what are the components, or what are the thousands of organic compounds which are present in the living tissues? So such kind of means what are those such kind of biomolecules? Simply we can be taken as metabolites. Metabolites. So means amino acids, so sugars, carbohydrates, polysaccharides, so protein, so whatever it is, definitely. So such kind of biomolecules we will be considering as metabolites. So these metabolites only we are calling by one more name. These are all called primary metabolites. These are all called primary metabolites. Simple my plants are there. So plants using some kind of organic compounds or some kind of biomolecules for the process of which one photosynthesis, and for the process of respiration, or for the process of light reaction, like next one electron transport system. Likewise, there are different types of processes taking place in the plants, even in human beings also, but plants I am taking as example. So there, what are the organic compounds involved in like chemical reactions? So such kind of biomolecules simply we will be considering as metabolites. These metabolites we are calling by one more name, primary metabolites. Which one? Primary metabolites. However, when one analysis, see there, when one analysis plant, fungal members or microbial cells, one would see thousands of compounds other than the primary metabolites. So, here nalla paatha adu enna. Suppose if you take, now in order to obtaining of either one particular chemical analysis, now we have taken the plants, or we are taking the fungal members, or we are taking the microbial cells. Microbial cells, bacterial members we can consider. So microbial cells we are taking. So one would see thousands of compounds. Again we will observe the thousands of compounds other than the primary metabolites. Not only metabolites, other than the primary metabolites, we will obtain, or we will see the thousands of compounds. These kind of compounds we will be considering as secondary metabolites. So thereby, did you understand one thing? Suppose if you take a living organism or one particular living tissue, in order to, or for obtaining of chemical analysis, definitely two types of metabolites we will obtain. One is primary metabolites and the second one is secondary metabolites. Point purida. Sir, what is the difference between primary metabolites and secondary metabolites, sir? Suppose if you take one living tissue, which one is a primary and which one is a secondary, how we can separate, sir? Did you get that particular down? Now you listen carefully. Just now one point save பண்ணிக்கோங்க. One is primary metabolites and second one is secondary metabolites. Means இப்போ whatever may be that living tissue, that is maybe plants or fungal members or the microbial cells or animal cells, whatever may be that living cell you can take there, definitely along with primary metabolites or other than the primary metabolites, there are thousands of compounds we will obtain. These thousands of compounds we will be naming as we are naming as secondary metabolites. Now once I will give you the examples of secondary metabolites, thereby you will understand that. Once you see alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gums, spices. So still so many, so I can keep etc etc so many things. Okay, so means here all these things we will be considering as secondary metabolites. Means here, so I think so you're understanding. So now by seeing which one rubber you see there, so most important things let us see, and colored pigments, scents, gums and spices you see there. So by seeing all these things, we will definitely understand one point, that is these are all the coming, or these are all excreted by the plant tissues or the animal tissues. So mostly so plant tissues only we let us consider. So thereby here rubber plants are there, and the next one scent plants are there, gum plants are there. Rubber plants, let us take example. இப்போ ஃபாரஸ்ட்னாலே போவோன்னா ஏதோ ஒரு பிளான்ட் இருக்குது அங்கே. ஸோ ரப்பர் பிளான்ட் இஸ் தேர் மீன்ஸ் people will go and they will put some kind of mark on the bark. So thereby after somewhat time the rubber milk will come out. I think so you can observe this one in Discovery Channel also. Next one, spices, we know very well. Gums, even camel gum or some kind of gum, or if you see their neem plant bark, some kind of gum it will come out. Point, did you observe? Later if you take some kind of plants, colored pigments. So if you take some kind of plants, there like so girls will wear like herbal henna madri we will use so that one, that leaf also if you keep on your palms or the kainale put one, so thereby by overnight that will be gives somewhat good color. So likewise colored pigments, scents we know very well, gums we know very well. Likewise, so all these kind of chemical compounds, our subject point of view we will be considering as secondary metabolites. For example, sapota, if you see there, like a pache sapota means raw sapota, if you would see there, if you pinch on that one, so definitely milk will come out, like a papaya. So in papaya also the secretion of milk we can observe there. So all such kind of chemical compounds simply we will be considering as secondary metabolites. Sir, why sir these secondary metabolites are producing in the plants means actually, so these kind of like a secondary metabolites are the products of chemical compounds. So suppose if one chemical reactions are going to be taken place in the plants, so these are all what are the metabolites or what are the chemical compounds produced after the metabolic reactions, so we will be naming as secondary metabolites. Okay, now while primary metabolites have identifiable functions and play a role in normal physiological processes. So definitely, so what is the information now we are getting? Suppose if you take primary metabolites, so we can identify their functions and what are the main important roles in the physiological processes we can identify. We can study, but once you see, we do not at the moment so understand the role or the functions of all the secondary metabolites in the host organism. So thereby, so what are the functions or what are the identical functions of the primary metabolites? So we can study very well, but we could not or we don't know so the effects or the role and the functions of secondary metabolites which are present in the host organism. Suppose suppose let me take one more time support example. So if you take raw support, no, there milk will come. So by seeing that, okay, it is raw one, it is not ripened, still likewise we will notice one thing. But when this particular milk is coming or producing, then what is the use of this one? Or papaya, there also some kind of like one more plants likewise. So there are different types of plants, but why these are all coming? What is the necessity? What is the importance of these chemical compounds to the plants? Likewise, we don't have any idea. So likewise, so author saying to us, however, many of them are useful to the human welfare. So means even though we are not having the information regarding the role or the functions of the secondary metabolites, so definitely we are ready. So definitely we are ready to use them for the human welfare point of view. So thereby here definitely you see there essential oil, alkaloids, flavonoids, rubbers, essential oils, antibiotics. So all these are all scents, spices. Next one, different types of colored pigments. All these are all chemicals or the main important roles in the plants. We may not know, even though we are using all these are all for the human welfare. So I hope you understood that particular point. Later, let us have a look on the, let us focus on the one important table which they have given in our NCERT. Once let us focus. So what is the information now? Here we can concentrate more and more means. Next one, now you see there some secondary metabolites have. So some secondary metabolites definitely have ecological importance. So means here secondary metabolites are useful to the human welfare, and some secondary metabolites have are useful for ecological importance point of view also. Okay, so thereby here ecological importance point of view, human welfare importance point of view, or human welfare purpose point of view, definitely we will use the secondary metabolites. So now here some small table is there. So here and here. So let us have a look on this table. Listen carefully. Is the table important? So till now, what is the information you understood? So whether you understood or not, forget it. But under primary and secondary metabolites, this particular table can be important for NEET examinations. Okay, so thereby here whether you will appear, whether you will write the NEET examination or not, that you keep it beside. But as a biology student, definitely you must remember, you must know this. Okay, so let us learn the one by one. Now pigments. Pigments, what are the pigments? No, carotenoids and anthocyanins. Carotenoids, anthocyanins are pigments. Next one, alkaloids, morphine and codeine. Pigments are carotenoids and anthocyanins. Alkaloids, morphine and codeine. Terpenes, monoterpenes and diterpenes. Next one, essential oils, that is lemongrass oil. Lemongrass oil. Now one more time you see there, pigments, alkaloids, terpenoids, essential oils are there. Now pigments are carotenoids, anthocyanins. Next one, alkaloids point of view, alkaloids are morphine and codeine. Next one, terpenoids point of view, monoterpenes and diterpenes. Next one, essential oils, lemongrass oil. Okay, next one, that side, on the side path come. Next one, toxins. Toxins are abrin and ricin. This is so many examinations they are asking. Toxins, abrin and ricin. Next one, lectins are concanavalin A. One more time, try to say concanavalin A. Next one, drugs, vinblastine, curcumin. Next one, polymeric substances are rubber, gum, cellulose. Right, now one more time, toxins, abrin and ricin. Lectins, concanavalin A. Drugs, vinblastine, curcumin. Next one, polymeric substances are rubber, gum and cellulose. Okay, shall we discuss by one more time? Let us learn from down to up in a descending order or ascending order, whatever it may be that. Now let us move. Polymeric substances, rubber, gums, cellulose, drugs, vinblastine, curcumin, lectins, concanavaline A, toxins, abrin and ricin, essential oils, lemongrass oil, terpenoids, monoterpenes, diterpenes. Next one, alkaloids, morphine and codeine, pigments, carotenoids and anthocyanins. My dear students, my request is, so totally once how many are there? Once you see 1, 2, 3, 4, 5, 6, 7, 8 only are there. Okay, so all these are all. So take or pass this video and learn this one. So then only you try to move forward. So up to here, detailed information we have discussed regarding primary metabolites and secondary metabolites. Primary metabolites and secondary metabolites. Now let us focus on the one more small concept, that is biomacromolecules. Biomicromolecules. Now there is one common feature, or there is a feature common to all those compounds are found in the acid soluble pool. Acid soluble pool or acid insoluble pool, likewise we have seen now. So means if you take one test tube, there acid soluble pool is present in, suppose at the time I have taken the example of tea also. So while filtering tea by mom, so she will pour the tea on the filter, that thing so there tea ulla poi, so what is a filterable thing is adi like a mesh mele da irukunu. So there, so here we have taken the tea as acid soluble pool, and what is a filter, means what is a tea powder will be there, that one we will be taking as acid insoluble fraction. So likewise here also one point you try to say. So that point is, so that point is, what is the point? Now there is one feature common to all these compounds found in the acid soluble pool. Found in the acid soluble pool. So thereby here one common feature is there. What are the compounds which are present in the acid soluble pool? So what is the common feature and what is the main important points? They have the molecular weight ranging from the 18 to 800 daltons approximately. Nulla point save pannikonga. So means what are the chemical compounds or what are the compounds? So whatever may be there, what are the compounds present in the acid soluble pool? Definitely having the molecular weight ranging from the 18 to 800 daltons. dA, the units of daltons. Simply I have written as a dA, but through my voice you can get it, daltons. So thereby ipo save pannikonga acid soluble pool nalle. So even though there are thousands of compounds are there, their definitely molecular weight will be 18 to 800 daltons da. Amma purida. Very good. Next one, if you take the acid insoluble fraction, acid insoluble fraction has only 4 types of organic compounds. Tea cup of filter powder is there, there likewise here also suppose edo one leaf adi grinder panni, so while grinding trichloroacetic acid, what is the thing we have added, so what is the chemical we have to add, adi add panni appuram, so filter pannavana, so 2 kinds means liquid, so ulle poi, so again debris LMA, so we will be, our subject point of view we are calling as acid insoluble fraction. So there, like a tea powder where the present will be there, likewise acid insoluble fraction also will be there or will be situated above the filterable filter mesh, isn't it? So likewise only has that particular acid insoluble fraction will have only 4 types of organic compounds. 4 types of organic compounds, that is proteins, nucleic acids and polysaccharides. Next one more is lipids. So now one more time, So 4 types of organic compounds, 4 types of organic compounds, 4 types of organic compounds there once you see. So what are the things over there? Proteins, next one nucleic acids, polysaccharides, and one more is lipids. Likewise, 4 important 4 types of organic compounds which are present in the acid insoluble fraction. Okay, so next one these classes of compounds with the exception of lipids. We are observing proteins are there, nucleic acids are there, polysaccharides are there, and lipids are there, we are observing. Okay, but here one exceptional point is there. Actually, we should not consider these lipids under insoluble fraction. So here one exceptional point is there. Why? Because here once you see these classes of compounds with the exception of lipids. So sorry, lipids over then why you have written under? So 4 types of compounds which are present in the insoluble fraction means reason I will give you. Listen carefully now, but not in this video. Next video I will give you. One point you see here. What is the point? I am going to extend to the next video. No, once you see acid soluble irukudana ange 4 types of organic compounds irukku. So what are those? Proteins are there, nucleic acids are there, polysaccharides are there, and lipids are there. Even here we have written even lipids also present in the acid insoluble fraction. But once you see here the compounds with the exception of lipids, again here exception is there. Sorry, exception is there means why you wrote here, you may ask. Why I have written on that explanation, so I will give you in next video. Wait up to that. Right, now let us continue. Now what are the components are present in the acid soluble fraction? Definitely have the molecular weight is ranges from the 10,000 daltons and above. So here what are the molecules are present in the acid insoluble pool? No, just molecular weight is 8 into 800 daltons approximately. And what are the molecules which are present in the acid soluble fraction? Molecular weight is 10,000 daltons and sometimes 10,000 above that. So thereby what are the components or what are the molecules present in the acid insoluble fraction? Molecular weight definitely more than 10,000 or 10,000 above. 10,000 and 10,000 above daltons. So now here these chemical compounds for our convenience to study. So these chemical compounds, what are the chemical compounds? Now we are observing all these kind of chemical compounds. Our convenience to study, now we are dividing it into 2 types. One is biomacromolecules and one more is biomicromolecules. Now one point you can give the answer so without explanation. So what is that? No, so that point, this biomacromolecules molecular weight is macro, 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 and ఇది micro. రెండుమే ఏ ఇంగా ఐదా డిఫరెన్స్. Macromolecule molecular weight 10,000 daltons and above. Micromolecule, can you guess here? 8 into 800 daltons. But sir, why sorry here lipids are not, even though we have written under acid insoluble pool, one exceptional point enna sir adena, now only I am saying. So here actually molecular weight of the lipids is below the 1000. But here we have studied what are the components which are present in the acid insoluble fraction. So ranges from the 10,000 daltons and above we have written, but one exceptional point is there under the lipids. The lipids just having molecular weight 1000 below. 1000 below means definitely that must be there in the acid soluble pool, but lipids are there in the acid insoluble pool. Point purida. So thereby here this particular point definitely you have to remember. So this particular point in tomorrow's class also, in a next class also I will focus. So nalla save pannikonga. So continuation information definitely I will give you in next video. Thank you.